Well, thank you and welcome to everybody. Thank you all for coming long distances and short. I think shortest distance is across the street and the longest distance is some 8,000 or 9,000 miles. So thank you everybody for being here. A special thanks to the local scientific organizers, to Cecilia Lindgren, Mark McCarthy, and Rory Collins, although he protests he did no work. To Rachel, to Ashley, to Amy, and to Arabella for actually this meeting come together. It's amazing because it came together on relatively short notice. I think the invitations went out only in September-ish, and everybody is here, and that's amazing. And again, big thanks to the Wellcome Trust for supporting this meeting here. So the origin of this meeting was a series of conversations amongst a number of us, notably Mark Daly, Judy Cho, and Nancy Cox, and myself, about the sense that we were at a turning point in the genetics of common disease. That, of course, there's been lots and lots going on, and there's always progress, but that things were falling together with a critical mass that said, well, it was really time to stop and ask about the next phase of human genetics. So I'll take a moment to just say a few words about the first phase of human genetics. Dates are somewhat arbitrary. I just picked 1985 to 2015 to just step back and think about what it was that had happened. And it was a mix of so many different things, so many different kinds of things from so many different people. But I'll just tick off what's gone on. Early concepts about mapping by linkage in affected families, really for Mendelian disorders with standard kind of genetic maps. Mapping by linkage disequilibrium in populations of what you could do with ultra-dense genetic maps. Then generating data, actually early genetic maps with a few hundred genetic markers that let you trace inheritance. Then this detail of a human genome project, bringing together the entire world to sequence the human genome, which was done as a great international consortium. But even before the Human Genome Project was done, a recognition that we needed more than the sequence of one human, but tremendous amount of genetic variation. And so a SNP consortium was organized. Then results started coming in, and linkage worked well with the ability to map hundreds, thousands of Mendelian disorders, but the standard linkage paradigms worked very poorly for common complex diseases. And one of the ideas was the notion that common disease was not simply a whole bunch of rare diseases mushed together, that the allelic spectrum might look quite different, and that common variants might play important roles. And if so, then linkage disequilibrium mapping could be a very powerful tool for it. It took understanding the human haplotype structure, and early studies that showed the structure of the human genome with regard to haplotypes led to another international consortium, the Haplotype Map Project. To make any of that practical, it took amazing tools like genotyping arrays that today can do millions of markers simultaneously. And then big roles of computational mathematical analysis, imputation, correction for population structure, and then results beginning to come in. Initial GWAS studies, a few hits and a lot of controversy about whether this was good for anything. And then large disease consortia, particularly credit to the Wellcome Trust Case Consortium, Case Control Consortium, and to many other consortia that grew up, and to Decode for the remarkable work it did. So this led to results, large-scale GWAS now with not a few hits, but many, many loci. And then back to analysis, heritability estimates in bulk. Peter Vischer and colleagues showing us that common variants actually explained an awful lot of heritability. Studies on genetic correlation between diseases, Mendelian randomization, polygenic scores. Then back to creating these amazing tools, massively parallel sequencing coming along that made it possible to think about exomes at hundreds of dollars, genomes at under $1,000, and those numbers will drop. And then visionary efforts to create whole national biobanks. I want to salute Rory and everybody in the UK for the UK Biobank and its model, including the free data sharing there, and then efforts for hospital biobanks. It's breathtaking when you think about what's gone on in 30 years and just look at all of that. And it was all of these different types of contributions. And it took a world working together. It took a world in conversation working together. The results have been, well, today more than 30,000 GWAS hits. Actually, if you really count unpublished things coming out of the UK Biobank, I think that number is getting close to about 100,000, but I'm going to go with the official statistic in the catalog for hundreds and hundreds and hundreds of diseases and traits, and already it's been revealing insights into common disease. So we have these nice pictures and these maps, and you can pick well-studied diseases, and there are hundreds of loci, which is up from roughly 0 loci not so long ago. Take schizophrenia, where the count was 0 and is now 245. And also with sequencing, rare variant association studies are beginning to find genes based on an excess of rare genetic variants. It's still slow and expensive. Most of the hits are in developmental disorders and intellectual disability from the DDD, the Deciphering Developmental Disorders Consortium, but we're now seeing hits in other diseases as well. And you could pick various diseases. I put a few that I'm fond of, where there are specific biological pathways and surprises and implications, say, in heart disease, where the genetics can tell you from first principles that HDL is probably not a very good genetic target, which has also been proven by spending several $billion, about $5 billion, to develop HDL drugs that have failed. And the genetics is perhaps a cheaper way to figure that out. Many, many, many such things of targets. But there are real challenges. With all of that body of science that I crammed onto a single slide there, that has gotten us to the point where for any trait we can identify many, many loci, going from loci to function, variance to function, or what I often abbreviate as V2F, is hard. Taking a bunch of samples, finding the variance by genetic mapping, then figuring out what are the genes affected by those genetic variants. What are the cell types in which they act? What are the cellular processes they impact? And then how do they affect function? Where by function, I'm going to go all the way to mean disease mechanism and therapeutic hypotheses. That's what we want. And I put a few examples of where through heroic efforts, people wrestled to the ground a locus. Now there's like 30,000 or 100,000 loci. And these heroic efforts are heroic. They're wonderful. I think we'll hear from people who will tell us about some of these, including how long it took to do. Sometimes 5 years to wrestle a locus to the ground. And I think the heroic people who did it would be the first to say they would like to do it a lot faster than this. And so the question that I think is ripe, that is in the air for the entire field, is how do we move from variance to function? We need many different complementary approaches all to be working. I have been struggling with different pictures to try to capture what's to be done. There is the linear thing. Get samples, find variants, identify genes, cell types, et cetera, et cetera. But what's becoming clear from the studies that people have done is that it's also possible to play leapfrog in places. And there are a variety of methods, and where there are methods, there are often data sets and tools that would make those methods powerful for all diseases. So I won't go through all the many arrows that I've tried to represent of things that either are being done or could in principle be done, but can't, but I'll point to a few of them. On that first line, more GWAS remains a good thing. It finds more loci. Fine mapping narrows down variance. And then the more GWAS is done across a lot of different studies, PheWAS, the ability to annotate a given variant with many different phenotypes, is powerful because it'll let us clump together variants into different groups that play roles in similar processes. Then to go from variants to genes, regulatory links. We'd ideally like maps that connect all enhancers to all promoters and all cell types, and we'd like maps of all EQTLs. We'd like to be able to find important variants by conservation, and increasingly by machine learning. Then we can leap sometimes from samples to genes through rare variant association studies, looking for strong alleles. But We need very large sample sizes for that. Then sometimes we can go straight from the list of GWAS hits to identify cell types without even identifying the genes, just because the GWAS hits are enriched in genes expressed in certain cell types. So heritability partitioning can allow us to leapfrog from the variant to gene to directly go to cell type. We also may be able to go directly to process by the same kind of enrichment that's there, if only we had a catalog of all cellular processes. And then, of course, we can go from variance all the way to disease prediction with polygenic scores. And then having figured out who's at risk for disease but hasn't developed it yet, we can go backward and study those people pre-symptomatically and figure out what processes are at work in those people even before they've developed the disease. So hopping around this picture with all different kinds of methods and all different kinds of tools and databases will be very important. Now, all of this is pretty generic stuff. Below the red line are disease-specific things, where you'd actually get in and do an honest-to-God experiment. Everything I described under the blue heading is just plain looking, computational looking with powerful databases if we built them. If you're willing to go to the lab bench, you could also do experiments. And then you can talk about finding allelic effects by CRISPR editing and CRISPR inhibition, RNA signatures, monitoring RNA signatures, protein function, genetic perturbation in cells, organoids, animal models, and of course, disease and process-specific assays, and we'll hear some about that. And then in this purple stripe below, directly studying patients in recall by genotype, either single locus genotype or polygenic signature, by physiological testing of those people recalled, and by clinical trials even in such people. That's kind of the things that I see going on. But what's it going to take to do it? You could say, well, people are beginning to do things like this. Now, every other time in human genetics that we've seen that there's people beginning to do things, we've recognized that it's so much more powerful if we come together as a community to define what needs to be done, define common efforts, define common data sets, standards. And so what will we need? There's clearly intellectual ferment right now. But the resources we're going to need, just to be straightforward, to study rare variants, we're going to need 100,000 plus cases of many diseases. It's clear what the numbers are. We're going to need them. So that shouldn't scare anybody. The numbers we have right now are inconceivable compared to where we started. And it's clear we could get to such numbers. Common variants, large biobanks with PheWAS, where we can look at how variants affect many, many different kinds of phenotypes. Well, I project, we've already got biobanks of 1 million plus around the world, either built or being built. I will project we're going to see 50 million people in biobanks in the not so distant future. The big issues will be data sharing around that. Cellular data. We have this human cell atlas being built, which is fabulous, and it's going to point to all the cell types. I suspect we're going to need cell atlas data on hundreds of people, initially in certain tissues, but eventually whole body, that will allow us to look at the genetic variation at single cell level across lots of people. We're going to need to infer complete catalogs of all cellular processes. Maybe we're going to machine learn and auto-encode and other things like that to be able to derive that catalog of all cellular processes so that we're going to recognize what different diseases do. We're going to need molecular tools. We're all enamored of the ability to CRISPR edit, but CRISPR editing one nucleotide won't do. We're going to need to CRISPR edit hundreds of nucleotides in parallel, read them out. We're going to be able to, I think we're going to need to do this with combined single-cell DNA and RNA, unless the CRISPR edits are made with absolute efficiency. We're going to need to recognize which cells we edited and what effect it had. Or to do any of those scientific things as a community, we're going to need a community. There are a whole lot of things which we need to solve together. Data sharing, issues around consent, privacy, data platforms, credit. We're going to need principles and practices. Now, don't get me wrong. This is not a heavy-handed thing. There are many sovereign nations working on these things, and many sovereign hospitals working on these things, and many sovereign laboratories working on these things. And the goal is not to impose anything. But there's an awful lot of power if we can come together and find reusable solutions and principles. And that can only be done as a community. Similarly, quality control. We've seen in human genetics that quality control has been tremendously important from the earliest days of sequencing the human genome to recognize where the data are not of high quality and need to be of high quality and how they can be, and also to quality control the methods. Then for scientific progress, as a community, we need to define shared projects that need to get done. Doesn't mean that everybody has to do them, but we have to identify with clarity, boy, if we only had X, Y, and Z, we could make a lot of progress. And it can't be everything. It's got to be, at any given moment, well-defined projects. The HAP map was a well-defined project for its time, and people thought hard about it. We need scientific forums to come together and just talk about progress. We need some kind of a place where we can monitor that progress. And we also need to think about pioneering the clinical uses. Within pharma, human genetics is still not completely routine, and it's not completely understood by all pharma management. We need to think about how to pioneer its uses in drug development. We need to think about how to pioneer the uses of human genetics in clinical patient care, where things like polygenic scores are by no means routine, and it's not even clear how one would really use them. So we want to promote progress, best practices, and alignment. In any case, this is why a bunch of folks thought it was time to pull together the community, because this was little more than just progress as usual and business as usual. It really was phase 2 of the genetics of common disease. So what's needed? I don't know. I mean, we wrote down a common disease consortium. I don't exactly know what I mean by that, but if I were to ask what's the purpose of the meeting, it's to come away figuring out what that means. This morning, over some caffeine, trying to get myself onto the time zone, I wrote an international consortium to accelerate the understanding of the genetic basis of common disease and its applications to medicine through transformative projects, tools, principles, and community. I'm not attached to those words, but I felt obliged to write down some words just as something to shoot at. I do believe it is the moment to create some kind of a common disease consortium, because across academia, industry, national healthcare systems, hospitals, we all are approaching the same problem, and either we're going to do it in a fragmented way or we're going to do it in conversation with each other. And that's the purpose of the meeting. I have no particular attachment to what this should look like, how it should look like, what its work should look like, but I'm pretty sure we need something like it. And my hope is before we all disperse, we will have that conversation and come away with some plans to turn this into a real communal effort. That's probably it. That's sort of the charge to the group from the organization. A bunch of us thought of doing this. We wrote to people who showed up in New York for a 25-person meeting in some actually nice hotel room with windows even in New York City. And at that meeting, it was concluded that it would be worth trying to pull people together for a larger meeting, and here we are. So that's it. Welcome. We're actually on time even, and I shall stop there. Maybe I'm not sure what we're supposed to do here, but I'm glad to take a question or so, but the question will largely be, I don't know. That's why we brought you all together. So.